For some reason, given the last section was about water and we said that life on Earth is carbon and water based, I get the feeling that it's not too much of a shock that this section is about carbon. So, carbon chains are the backbone of biochemistry. Yeah, okay, you can have some nitrogens and oxygens mixed in, you can sometimes have sulfur and phosphorus, and you can sometimes have some other stuff as well. But carbon and hydrogen are the main components of biochemistry. So the simplest hydrocarbon, which is a chemical that's made of only hydrogen and carbon, is methane. And then we have ethane, propane, butane, and so on. Now it has one carbon atom bound to 4 hydrogen atoms in the case of methane. If you have 2 carbons, what happens? Well, each carbon can make 4 bonds. So you can have this. Or what else could you have? Well, you could potentially have a double bond. This is unsaturated. Whereas having as many hydrogens or other groups as you can fit, i.e. no double bonds or triple bonds, is saturated. So, what's so important about unsaturation? Well, addition reactions, which are involved in the biosynthesis of many compounds, use double bonds or triple bonds quite often. Now here, this is not an addition reaction. This is a dehydration reaction if you have oxygens here. Why? Well, hey, we're going to have OH and OH as substituent groups instead of hydrogen. So this would lead to a dehydration or condensation reaction where water is lost. So you lose OH from one side and H from the other side, and you bridge with an O. That's pretty much what starch ends up being like if you add the appropriate hydroxyl groups all over this in the case of a hexose sugar. This is not a hexose sugar because it doesn't have the necessary oxygens involved. Okay, why is a carbohydrate called a carbohydrate? It's because the fundamental formula for a carbohydrate is this to some multiple. What do we call this formula? This is called an empirical formula. It's representing the lowest subunit that can be considered a carbohydrate. We notice that it's carbon and water, hence carbohydrate. Okay. Now, carbon can form bonds with itself or with other atoms. So what happens when we have some other atoms? So a functional group or substituent group sometimes. So functional groups participate in chemical reactions. And they can include the hydroxyl, as found in alcohols, and sugars. Hydroxyls are what allow the condensation reaction that forms starch from glucose subunits to occur, or cellulose, which is a different conformation of bond, hence why most animals cannot digest cellulose. Basically all animals are incapable of producing cellulase, with the possible exception of, say, silverfish, but we can have symbionts that digest it for us, like, say, protists, or bacteria, or archaea. So it could be foregut fermentation or it could be hindgut fermentation, depending on the animal in question, but that's something we can cover at a later point. Now, carbonyls or carbonyls can be aldehydes, so they can be at the end of a carbon chain, or they can be ketones, so not at the end of a carbon chain. They have somewhat different chemical properties depending on whether they're at the end or not. For example, if you oxidize an aldehyde, you can produce a carboxyl. Carboxylic acids have carboxyl groups, and they can ionize, so the proton can come off and you're left with COO minus. You often see this written as COOH, just like that. Now, the amino groups characterize a group of chemicals called amines. They could just be NH2 attached to a carbon. So basically, let's take one of the hydrogens off of ammonia and substitute in the carbon chain instead. Or we could have this, say like this. So carbons over there and hydrogen. You will notice that in skeletal diagrams, this, for example, is butane. 4 carbons. And if we wanted to add 2 more carbons, there we go, hexane. So, when we draw something like this, that implies that there's carbon, carbon, nitrogen, and we note the extra hydrogen. Or we could have another form of amine that's like this. So we have primary amines, secondary amines, and tertiary amines. All right, so phosphates. What are phosphates used for? Well, they're charged. This can be used for, say, binding to positively charged things, potentially. Or we can just use this to make one side of some molecule hydrophilic, whereas the other side can be hydrophobic. One of the reasons why DNA is stable is because the insides are more hydrophobic than the outsides. The phosphate groups happen to be on the outer backbone, the sugar phosphate backbone. Is this a coincidence? No. Okay. Phosphates are also used for many other cellular molecules. What do you think happens if you string a bunch of phosphates in a row? The negative charges will try to push each other apart, right? So when you do that, that's a high energy bond. It will release quite a bit of energy when you let it fly apart. So, ATP, adenosine triphosphate. This should be a term we are reasonably familiar with. It is the main energy carrier molecule in, well, cells on Earth. Yeah, okay, there's also GTP, guanosine triphosphate, and CTP and so on, but that's another story. Those are used much less than ATP. So, in ATP, we can break the last phosphate off, or we can break the last 2 phosphates off. And what happens? Energy is released because of essentially the stored energy in having all these negative charges crammed together. Okay, sulfhydryl groups, or thiols, when you name them, they start with mercapto, usually. And, well, that actually depends on priority of naming groups. But regardless, sulfhydryls or thiols are responsible for, say, welding different parts of proteins together. How do they do this? Well, the disulfide bridge is pretty resilient. It holds up well in oxidizing conditions. In reducing conditions, though, the 2 cysteines that are on either side will end up with just SH instead of this disulfide bridge. When you have oxidizing conditions, the H's get ripped off, and this bridge forms. And thus, a protein that has disulfide bridges as a necessary condition for its function Generally, you're going to find that in relatively oxidizing environments. Now, some other characteristics of functional groups, well, we have the ionic functional groups, so carboxyl, amino, and phosphate. Phosphate, well, the isolated form of a carboxyl would be a carbonate. Of amino, it would be ammonium or ammonia. And for phosphate, you'd end up with phosphoric acid. You'd probably seen this before on, say, pop bottle ingredient lists. So yeah, pops are not healthy, but they do contain an essential nutrient, if you decide to look at it that way. Okay. So the carboxyl group functions as an acid. It will release a proton. The amino group will pick up protons, and the phosphate group releases protons. Now, generally, the phosphate group exists in solution as a charged form because of its pKa's, which are a concept that gets communicated better in chemistry class. So the acid dissociation coefficient. Okay, so other than the ionic ones, we can also consider the polar ones. For example, the alcohol group, so the hydroxyl, is polar. So are aldehydes and, to a lesser degree, ketones. Why to a lesser degree ketones? Well, something like acetone, which you can represent using this skeletal structure, that's not very polar compared to, say, an aldehyde. Generally, aldehydes are a bit more polar than ketones of equivalent mass and size, of course. Because, I mean, you could try to give me 10 carbons in a row with an aldehyde group versus acetone, the simplest ketone. Of course, that's not a fair contest. Okay, so one of the main reaction types that we're going to look at in biology, as opposed to chemistry where we kind of have to look at all of them, is dehydration and the associated counterpart hydrolysis. Dehydration, water gets lost. Hydrolysis, well, water is shoved in, hydro, and lysis is to break apart. So it breaks things apart. Now, when you use dehydration, you are joining pieces together to form larger molecules. Whereas if you use hydrolysis, you are splitting larger molecules into smaller pieces. Both are very important. And, well, this here, this synthesis of larger molecules, that's generally associated with anabolism. Anabolism is a part of metabolism where things get built up. Anabolism. Build things up in terms. And catabolism is breaking things down. Break things down. Okay, now in summary, carbon atoms form the backbone of biological molecules. They can link together to form chains, branches, and rings. Now, whether these rings have some special properties that are very, very distinct from chain forms, as the benzene ring does, or whether they don't, that depends on their specific configurations. Now, functional groups have polar or ionic qualities. These influence how they interact with water and other molecules. So in other words, if the functional group is polar, great, they will tend to interact with water better than less polar ones. If they're ionic, great, they interact with water even better. If there's a lot of these polar or ionic functional groups, the molecule overall is probably hydrophilic, so it will dissolve readily in water. It has greater affinity for water. If you don't have these functional groups that have good affinity for water, no, it won't dissolve well in water. Now, during a dehydration reaction, you remove water from subunits. This could be, say, a carboxylic acid and an alcohol, or it could be two alcohols. Now, for water, what actually happens is that this is the reaction that occurs because of the specific mechanics, namely substitution. We can talk about nucleophilic versus electrophilic substitution in chemistry class instead. Okay, during a hydrolysis reaction, that's the exact opposite of dehydration. So larger molecules react with water, which basically gets shoved in, and they break apart to make smaller subunits. So both of these have their place in biology because, well, the proteins didn't come out of nowhere. They had to be synthesized from amino acids. But where do the amino acids come from? Well, potentially, they could be synthesized in place, pretty much, or they could be from diet. So we have to eat proteins, break them down into amino acids, and reassemble them to make proteins that we need. Yes, I do realize that plants are somewhat different, but we'll put that aside for now. Even they have to recycle old, used up, broken proteins to be more efficient, because otherwise they lose some of their photosynthetic yield, which is not such a good thing, even though we all know that plants do sacrifice some of their yield so that they can reproduce better. For example, toxic plants, yeah, that's sacrificing some of their metabolic output so that they can have a better chance of surviving to reproduce, or reproducing successfully, as the case may be. Well, that's it for this section, and next section we can talk more about the basic chemistry of life, now that we've talked about water and carbon, so the basic building blocks.